Bismillahirrahmanirrahim. Students, today we will discuss the structure of monocot stem. Monocot stem shows the following major parts. The outermost layer is the epidermis. Below the epidermis is the hypodermis, which is the second part of the monocot stem. The third major part is the ground tissue, which extends from below the hypodermis towards the center of the stem. And then the fourth major part are the vascular bundles, which are present in scattered form in the whole ground tissue. Now we will discuss each part independently. In this monocot stem, we will study in detail the anatomy or internal structure of monocot stem of maize plant. The outermost part, which is epidermis, it consists of single layer of cells which are compactly arranged with no intercellular spaces, and on the outer side of the epidermis a cuticle layer is present and the epidermal hairs are absent and stomata may be present. The second part next to epidermis is hypodermis, which consists of 2 to 3 layers of sclerenchyma cells, and this region is called the hypodermis. Sclerenchyma may be interrupted, means that in between the sclerenchyma cells where stomata are present, just below these the parenchyma cells may be present which consist of chloroplast, and that region is also known as the chlorenchyma. The third major part is the ground tissue system, which extends from hypodermis to the center of the stem, and ground tissue in monocot stem, it is not differentiated into cortex, endodermis and pericycle and pith as in dicot stem. And this region in monocot stem, it consists of thin-walled parenchyma cells with intercellular spaces. Then in the ground tissue, the scattered vascular bundles are present, which form the fourth major part of the stem. Vascular bundles in the monocot stem, these are conjoined, mean that xylem and phloem, these are present in the same bundle. Collateral mean that xylem is present on the inner side of the phloem, and closed means that vascular cambium is absent in these vascular bundles. Means that in monocot stem the vascular bundle, they show three main characteristics. Number one, these are closed, these are conjoined, and these are collateral. The bundles are scattered in the ground tissue, and the bundles which are present on the peripheral part, on the outer part of the stem, these are more in number but small in size, while the vascular bundle which are present towards the center, these are less in number but these are larger in size. And each vascular bundle is surrounded by a sclerenchymatous sheath, which is also known as the bundle sheath, which is made up of the sclerenchyma cells, and this sheath is more prominent on the upper and the lower side of the vascular bundle. In the vascular bundles, xylem and phloem are present. Xylem is Y-shaped. The arms of the Y-shaped xylem are formed by the pitted and bigger vessels, which are known as the metaxylem, and the tail of the Y-shaped xylem is formed by the smaller annular and spiral vessels, which are the protoxylem. And in between the metaxylem vessels, pitted tracheids are also found. On the inner side of the protoxylem, a cavity is present, a lysogenous, which is known as lysogenous water cavity, and it is formed by the breakdown of protoxylem vessels, and around this cavity wood parenchyma or xylem parenchyma is present. The phloem in the vascular bundle is present above the xylem, and it consists of sieve tubes and companion cells. Phloem parenchyma is absent. The protophlloem is present on the outer side and it is pushed outside and it is crushed and it becomes non-functional, while the inner portion of the xylem phloem, it is functional and it known as the metaphloem. Now we see the diagrams showing the major parts of the monocot stem. This is the general diagram of the monocot stem, in which the outermost layer epidermis, and below this epidermis hypodermis is present, and this is the ground tissue. In this ground tissue the scattered vascular bundles are present, and in these vascular bundles we can see here that on the outer side the size of the vascular bundle is small and these are large in number, while in the central part the number of vascular bundle is small. These are less in number and the size of this is small, large. And in these vascular bundles, each vascular bundle, the xylem and phloem is present. Phloem is on the outer side, while xylem is present on the lower side. We will see in detail these diagrams. In this diagram, the part, a small portion of the transverse section of the monocot stem is shown here, in which the parts are visible. The outermost cuticle, below the cuticle layer epidermis is present, and below the epidermis hypodermis is present. And in this hypodermis, below the stomata, parenchyma cells are present, which contains chloroplast. So this region is known as the chlorenchyma in the hypodermis. And then below the hypodermis, the ground tissue is present towards the center, and this ground tissue, it consists of parenchyma cells, and in this ground tissue vascular bundles are scattered. This is the detailed diagram of vascular bundle of the central part, which is large in size. In the next diagram we can see the detailed structure of the parts of the monocot stem. The outermost layer is epidermis. This is the epidermis, which consists of compactly arranged parenchyma cells which have no intercellular spaces, while below the epidermis 2 to 3 layered region is present, which consists of sclerenchyma cells, which forms the hypodermis. And then below the hypodermis towards the center, this is the ground tissue, which consists of the thin-walled parenchyma cells of different sizes, and in these parenchyma cells, parenchyma tissue, in the ground tissue, the intercellular spaces are present in between the parenchyma cells, which are the intercellular spaces. These are prominent in the ground tissue parenchyma cells. In this ground tissue, vascular bundles are scattered. The outer parts, peripheral parts vascular bundle are small in size and large in number, while the central part of the ground tissue, it contains large size vascular bundle, but the number of these vascular bundle is small. And here we see the detailed structure of the vascular bundle. This is the bundle sheath which is present around the vascular bundle. made up of the sclerenchyma cells. So it's also known as sclerenchyma sheath or bundle sheath, and this sheath is more prominent on the outer side of the vascular bundle and the lower side of the vascular bundles. Then above the xylem, phloem is present, and this is the crushed phloem, which is formed by the protoxylem cells, phloem cells, and below the protophlloem cells, metaphlloem cells are present, which are functional. And then below the phloem, xylem is present, which is Y-shaped. Arms are formed by the metaxylem cells, and the tail is formed by the protoxylem cells. And below the protoxylem cells, lysogenous cavity is formed, which is formed by the breakdown of the protoxylem cells. In the next diagram, which is colored, we can see more clear picture of the different parts of the monocot stem. Cuticle outermost, epidermis, then below epidermis, hypodermis region is present, which is made up of the sclerenchyma cells. And in this hypodermis region, in some parts where stomata are present, below stomata, the parenchyma cells are present, which contains chloroplasts, and then this part, a parenchyma cell which contains the chloroplast, is known as the chlorenchyma. Then below the hypodermis, ground tissue is present. We can see here the parenchyma cells with intercellular spaces in between these parenchyma cells, and vascular bundles of the outer region, small in size and more in number, and the vascular bundle of the central region, which is large in size, but the number of vascular bundle is small in the less in the central region of the ground tissue. Bundle sheath, we can see bundle sheath, prominent bundle sheath on all sides, but more prominent on the upper and the lower part of the vascular bundle. Phloem, in which the crushed phloem is present toward the outer side, formed by the protophloem cells, and this lower functional part of the phloem is the metaphloem, and this phloem consists of sieve tubes and companion cells. This is Y-shaped xylem. The arms of the xylem are formed by the larger pitted vessels, which are called the metaxylem. Means the thickenings which are present in these cell vessels are the pitted, while the tail of the protoxylem, xylem is formed by the protoxylem cells, which are small in size and consists of annual and spiral thickenings. The walls of the protoxylem cells have annual and spiral thickenings, and then below this protoxylem a cavity is present, which is also known as lysogenous cavity, lysogenous water cavity, which is formed by the breakdown of the protoxylem cell, which are present below these protoxylem cells. And then around this vascular bundle sheath is present. In between the metaxylem cells, pitted tracheids are also present, and around this water cavity, lysogenous water cavity, wood parenchyma or xylem parenchyma cells are also present. So students, today we discussed the structure of the monocot stem of maize plant, which consists of the main parts. Number 1, epidermis, which is outermost layer. Below epidermis is the hypodermis, which is sclerenchymatous, and then consists of 2 to 3 layers of cells. Then below the hypodermis, the third major part is the ground tissue, and this ground tissue is formed by the parenchyma cells with conspicuous intercellular spaces. And then the fourth major part is that in the ground tissue scattered vascular bundles are present, and these vascular bundles on the peripheral side are more in number while small in size, but the vascular bundles in the central part are large in size but less in number. I hope you have well understood the diagram and the structure of the monocot stem of the maize plant, and in the next lecture inshallah we will discuss the structure of the dicot stem.